Alright guys, welcome back to the channel. So today we'll cover the second chapter of the biology syllabus, which is biological molecules. And in this chapter, we'll talk about the structure of carbohydrates, proteins, and lipids. And we'll talk about structures in depth that we need to know, for example, cellulose and others more. We'll talk about them more in detail. Okay, so let's start. So the first thing that we need to know are important terms which are used in this chapter. For example, macromolecule. So it basically means that it's a large biological molecule. For example, a protein. A protein is basically a polypeptide and it's made from a monomer. So we'll come to what a monomer is later, just like right after this. And then it can also be a polysaccharide or a nucleic acid, which is DNA or RNA. So a monomer is basically a simple molecule. It's a smaller molecule which acts as a repeating unit for synthesis of larger molecules. And this is by condensation reaction. So we'll talk about that as well later. So monomers of a protein is peptides or amino acids. So they form peptide bonds and then it becomes a polypeptide. And then, yeah, so that's how a protein is formed. And a protein can be made up of multiple polypeptides. So a polymer is a giant molecule, which is also made by similar smaller subunits known as a monomer. So the difference between a polymer and a macromolecule is that a polymer is just straight up made by monomers, and macromolecules can be comprised of more than one polymer or different parts of it. It's more complex in structure. So the first thing that we need to talk about is carbohydrates, and we've already covered this in our previous syllabus. And the first subunit under carbohydrates is monosaccharides. So monosaccharides are basically a single sugar molecule, while terms like disaccharide means that there's two sugar molecules. So they follow a certain general formula of CH2O and the whole thing bracket N. So this shows that the H and O is always in a ratio of 2:1, and it can be either triose, pentose, or hexose. So it's a 3-carbon, 5-carbon, and 6-carbon respectively, and the 3 pent and hex is referring to the number of carbons present in that sugar molecule. So they're usually used to either synthesize larger molecules, or they can basically be used for metabolic reactions. So for example, glucose is used in aerobic respiration. So we'll talk about more in depth about the structure of glucose. So glucose does have a straight chain structure, but it's mostly represented by a ring structure. So it's a lot more stable in the ring structure. So this is the ring structure of glucose. So glucose, when it forms a ring structure, it can form either alpha or beta glucose. So the ring structure of both of these glucose is almost the same, but this carbon right here, you have to look at the position of the OH bond. And this will make a big difference when you talk about how glucose is used to make larger polysaccharides. So about starch and glycogen compared to cellulose. So we'll come to that in a bit. So in alpha glucose, the main thing that we need to know, right, is that this OH bond is below the ring structure. And for beta glucose, this OH bond is above the ring structure. So you do need to memorize these drawings or these structures. So I would recommend you to draw this out in your notes or in your textbook, or sorry, in your notes basically. And then they're important because we'll need them to draw larger polysaccharides. So for drawing 1,6 glycosidic bonds and 1,4 glycosidic bonds. So you should take note of these structures. So disaccharides, like I said, they are a sugar molecule that contains 2 sugar molecules. So they're formed by glycosidic bond, which is between 2 monosaccharides. So maltose, for example, is made by glucose and glucose, while sucrose is glucose and fructose, for example. So formation of a glycosidic bond is basically a condensation reaction because water is produced as the other product or is eliminated during the reaction. So let's look at this structure here. So you have 2 alpha glucose molecules. So just now we discussed that maltose is a disaccharide which is made of 2 glucose molecules. So when we draw the ring structure of glucose, we represent that the OH bond is below the ring. And then this position here, so these numbers here represent the carbon. So for example, this carbon right here is in position number 1, 2, 3, 4, 5, and this last one is 6. So this carbon on number 1, it will lose its hydrogen, while this carbon on number 4 of the next glucose molecule will lose its OH. So an H2O molecule is eliminated or produced in the reaction, and then this oxygen atom which is left, it will connect these two glucose molecules and form this glycosidic bond. And this is said to be a 1,4 glycosidic bond, and 1,4 basically refers to the position or the carbon which forms the glycosidic bond. So if you take note of the first glucose, it's the first carbon, and then for the second glucose, it's the fourth carbon. So let's move on. So you also need to know the structure of sucrose. So it's formed from alpha glucose and also beta fructose. So you need to know the structure of beta fructose. So we know that the structure of alpha glucose, which is on the left-hand side, with the OH bond below the ring. So there's also the structure of sucrose, which is also a 6-carbon compound, but then the two ends has this CH2OH groups. Okay, so basically also forms another glycosidic bond by elimination of the OH and H. So it's CH2OH here, and then hydrogen will be attached to this other carbon. Okay, and this is basically a condensation reaction. So if you want to go back from sucrose to produce alpha glucose and beta fructose, that's known as a hydrolysis reaction, because H2O is used as a reactant in that reaction to break bonds. So this is what we have learned in our previous syllabus on how we test for reducing sugars. So all monosaccharides and some disaccharides, except for sucrose, they are known as reducing sugars because they can undergo a redox reaction. So this is for you to understand why this reaction occurs completely, although you won't be really tested on this. So the Benedict solution is a solution which contains Cu2 plus ions. And then when this reaction occurs, Cu2 plus will be reduced to form Cu plus, or the copper oxide, during the reaction. And the copper oxide is the origination of this insoluble brick red precipitate, which shows that a reducing sugar is present. So how do we do the Benedict's test? So we add excess. So just take note that you have to add excess Benedict's reagent to a solution which contains the sample, and then you put that entire test tube in a water bath and you heat it. So the color change is from blue. Blue indicates there's no reducing sugar present. And if there's any color change in this intensity order, green, yellow, orange, and red, brown, it'll form a precipitate, but it'll be of different colors. So intensity of color will indicate the presence of how much sugar is present in the sample. So you can also use a colorimeter to measure the color change more precisely. This is helpful in paper 3 when they ask you what adjustments you can do to a specific experiment. So you can suggest colorimeter if there's anything related to color. And this color change is usually tested in MCQs. So it's good to remember that it follows the order of green, yellow, orange, and then red, brown being the most highest concentration of sugar, reducing sugar. So we know that we can test for reducing sugars, but we have not yet learned about how to test for non-reducing sugars. So as I just said, sucrose is a non-reducing sugar. So when you put Benedict's test, it will remain blue, which means that there's no presence of reducing sugars. So how do we test for non-reducing sugar? So it's a disaccharide in the first place. So you can do a hydrolysis reaction, which means you use HCl to break the glycosidic bond. And then once you break the glycosidic bond, it will break into its two monosaccharides, right? So then you can add the Benedict's reagent. So you add excess Benedict reagent to the sample and then use NaOH, because Benedict solution can only work in alkaline conditions. So then you can basically see blue is no reducing sugar present, to a color change, like I mentioned here. So green, yellow, orange, or brick red PPT. And if there's a color change, non-reducing sugar is present. So the next thing you need to know is polysaccharides. So these are made from monomers or subunits, and the subunits are monosaccharides. And they usually involve condensation reactions or formation of glycosidic bonds between the monosaccharides. And glycogen and starch, they are made from alpha glucose, and cellulose is made from beta glucose. So let's talk about these structures more in depth now. So why do we need glycogen and starch in the first place? So we know that glycogen and starch act as storage molecules for glucose. And the main issue comes from the fact that glucose is soluble. So it will affect the osmotic balance. So for example, if there's a high concentration of glucose, it will reduce the water potential. And glucose is very reactive, so it will affect the cell chemistry. So these problems are avoided by converting it into polysaccharides. So these polysaccharides, glycogen and starch, first of all, they're inert. So there's no issue of affecting cell chemistry, insoluble, so they do not affect the osmotic balance as well. And they're also very large molecules, so they won't be lost through the cell surface membrane. Okay, let's talk about the structure of starch and glycogen. So they are, first of all, polymers of alpha glucose. So starch is a mixture of amylose and amylopectin. Amylose is made by condensation reactions between alpha glucose molecules. So alpha glucose is the glucose is a monomer for this, and it forms long unbranched chains of 1,4 linked glucose. So as I said before, 1 and 4 refers to the carbon on the ring or the position of the bond. And amylopectin, it's made of many 1,4 linked alpha glucose, and also at branches, where branches are formed, that's a 1,6 linked alpha glucose. So let's look at an image here. So these straight chains, they are 1,4, but then when there's a branch forming, it's 1,6. So let's look at how the structure is drawn when you just take note of the rings. So we already know what a 1,4 is. So between here and here, so loses H2O, forms a glycosidic bond here. And over here, the OH, which is below the ring, and then over here, CH2OH, this will lose. So this H will be gone, and OH, water is produced, and this is a 1,6. So 1, this is the position of the first carbon, and position of 6 carbon. Okay. So the next thing is glycogen. So it has a similar structure to amylopectin, and it is also branched, so it contains both 1,4 and 1,6 bonds, but it's much more branched than amylopectin. So what is the adaptation of it being so branched? So when glucose is required by the body, so multiple enzymes can hydrolyze the entire glycogen molecule from multiple ends. So more glucose is released, and it can be released into the bloodstream faster. So now we're going to talk about the structure of cellulose. So first of all, take note that the cellulose is a polymer of beta glucose, unlike the previous two we talked about. So cellulose is present in cell walls. So we have always been talking about cellulose cell walls. And it's a structural molecule. Sorry, this is not meant to be a wall here. So it's a structural molecule because it's a mechanically strong molecule. It has a structural role. So the OH bond in the beta glucose is above the ring, as we have learned. So to form an unbranched long chain, every beta glucose molecule must be rotated by 180 degrees. And then arrangement of these beta glucose molecules, rotating it by 180 degrees, will result in formation of hydrogen bonds between the H and O, which is of the OH groups present along the cellulose molecule, so the long molecules. And then these cellulose molecules, right? So 60 to 70 of them will clump up together because of this hydrogen bonding and will form a bundle, which is known as a microfibril. And then microfibril bundles itself will also be held together in bundles. And then this will form a fiber. So a large number of hydrogen bonds is formed between cellulose molecules, between microfibrils, and this gives the high tensile strength and allows it to be a mechanically strong molecule. So this is what I just said. So large number of hydrogen bonds between cellulose molecules to form microfibrils, and also between microfibrils to form fibers, high tensile strength. So tensile strength basically means that how much energy it takes to break it when it's stretched. So if it has high tensile strength, it can withstand stretching. So it allows plant cells to take in a lot of water. So in the last video, we talked about in the cell surface membrane why plant cells won't burst when there's a large influx of water into the cell, because the cellulose molecules or the cellulose cell wall can withstand the outward pressure by water. Okay, so this is the cellulose molecule. So you'll have to rotate this structure by 180 to bring this OH bond up so that this glycosidic bond can form. So if you notice when you draw the full multiple repeating units, so you'll have this one bond up here, and then now that this bond is here, this will form here. So basically, the glycosidic bonds will flip up and down. So 1, 2, 3, 4. Okay, hope that makes sense. So do take note of these structures because they can ask you to draw them, especially cellulose or 1,4,1,6 glycosidic bonds, and especially these structures. It's quite difficult to think of and come up with in the exam if you have not studied it well. Okay, so let's talk about lipids next. So lipids are basically organic molecules which are defined to be insoluble in water. So lipids, they have in lipids we talk about fatty acids first, and fatty acids have a carboxylic head or carboxylic group on it as its head, which is a COOH group. So a carboxylic acid group, as we have studied in chemistry, and the remaining part of the fatty acid is basically a long hydrocarbon tail. So it has many CH2 bonds and ending with CH3. And along the fatty acid tail, so as we talked about this in the cell surface membrane chapter, there can be multiple C double bond C along the tail, which are known as kinks. So it basically forms an unsaturated fatty acid. So unsaturated means that it does not have the maximum number of hydrogen which can be bonded to it, right? Okay, so this is a fatty acid. So this is a carboxylic head or carboxylic group. And then you see CH2, this is a long hydrocarbon tail. And then this is a double bond. So it doesn't have the maximum number of hydrogens bonded to the carbon. So monounsaturated means there's presence of one C to C double bond C bond. Polyunsaturated means there's multiple. And this COH group is important because they can form ester bonds with an alcohol. And most importantly, we talk about glycerol because it'll be important in formation of triglycerides. And ester linkages is basically C double bond O bonded to an O next to it, adjacent to the carbon. And this is also a condensation reaction where one molecule of water is produced. So the structure of glycerol is basically, it has 3 hydroxyl groups. So hydroxyl groups are basically OH bonds. So it has 3 OH bonds. So it can bind to 3 fatty acids because of one fatty acid has one COH group. So 3 water molecules are produced at the end. Okay, so this is the structure of a glycerol. So CH2OH, CHOH, CH2OH, which means that there's 3 OH bonds along the molecule. So triglycerides, they're insoluble in water, but they're soluble in organic solvents like ethanol. So this allows us to test for lipids, which is the ethanol emulsion test. We'll talk about that in a bit. And because hydrocarbons are long and mostly unpolar, so there's also uneven distribution of charge. So it's not polar basically, so it cannot really form bonds with water, which is what gives compounds their solubility. So their function is mostly acting as energy reserves because they have more CH bonds, and more CH bonds, which means that they will yield more energy on oxidation. They can also act as an insulator under the skin, for example, blubber in whales. It also provides buoyancy for blubber specifically. And so it acts as a source of metabolic water. So metabolic water is basically when water is produced as a product, and that water can be used for reactions or whatever is required, because especially in dry areas like where there's not a lot of water, so water can be produced by metabolic reactions. So we have already covered phospholipids in the cell surface membrane chapter, so I'll just quickly go through it once again if you didn't understand. So what we know about phospholipids is that it has a head which contains a phosphate group, and only 2 ester bonds are formed during this formation of a phospholipid, and it has only 2 fatty acid tails, or 2 hydrophobic fatty acid tails. And the heads, they're hydrophilic, and tails, they're hydrophobic, so they can form a bilayer in the cell surface membrane because the hydrophilic part will face the water and hydrophobic part will face inwards and towards the other tails. Okay, so let's talk about the test for lipids. So we just learned that lipids are soluble in ethanol but not in water, so we can do the ethanol emulsion test. So first we obtain the lipid. So if it's a solid sample, you'll have to crush it to obtain the lipid. So you can use a mortar and pestle, for example. and then you put it into a test tube with absolute ethanol. Absolute ethanol means there's very little water inside, and then you shake it. So the lipids will dissolve, and then the test tube is decanted into another test tube containing water. So decanting means that you just pour out the content of the test tube into another test tube. And if lipids are present, a cloudy white suspension is formed. So if no lipids are present, the ethanol will mix with water. But if lipids are present, ethanol cannot dissolve. So a precipitate of some sort can be seen. Okay, so the next part is about amino acids. So the most important thing that we need to talk about in this will be about proteins, because proteins can form enzymes, and we have already studied about enzymes. But the most important thing about enzymes is that all enzymes are proteins, but not all proteins are enzymes, because proteins can have structural roles as well. So we'll talk about that when we talk about hemoglobin and what's that compound? Collagen, sorry. So proteins can also be hormones, insulin, glucagon, and antibodies. So this is important. Just take note they're antibodies for now, because they do ask this in MCQ. So proteins' most basic structure is amino acids. So the structure of amino acids is that they have an NH2 group on one end and a COH group on the other. And the middle part is bonded to a hydrogen and an R group. So an R group can vary. So for example, glycine's R group is just a hydrogen atom, while this another amino acid called aspartic acid, it is a CH2COH group. So R groups will determine if the entire amino acid is polar and also its overall charge. So polar amino acids will have groups like COH and NH2, so they can be ionized to form COO minus and NH3 plus. And pH is determined by the overall charge. For example, if it has two COH groups, it'll be overall negatively charged and will be somewhat acidic. So proteins have multiple structures. So we have a primary structure, secondary structure, tertiary, and quaternary. So if quaternary talks about two or more polypeptides, etc. So we'll come to that now. So the primary structure is basically the sequence of amino acids, and amino acids are joined together by peptide bonds. So this is also a condensation reaction because water is produced. So this COH group and this NH2, right? They will react to form a CONH, which is an amide linkage, and OH is removed from COH and H is removed to form water. So this is a dipeptide. So two amino acids can bind together to form a dipeptide. And if you add more amino acids into the chain, it will become a polypeptide. So as I was saying again about the structure of amino acids, so one group has the NH2, the middle part CH, and this is the R group, which can vary between amino acids, and then the other part is the COOH group. So this is the somewhat general structure of amino acids. Okay. So as we're talking about primary structure, we know that it's a specific sequence of amino acids due to formation of peptide bonds, and every different type of protein will have a different primary structure. The secondary structure comes from regular coiling and folding. This is mostly because of hydrogen bonds to form an alpha helix or beta pleated sheet. And tertiary structure is also due to 3D coiling of an already folded chain of amino acids due to multiple different types of bonds, which is hydrogen bonds, disulfide bonds, ionic bonds, and hydrophobic interactions. So what we have first is that multiple amino acids will join together to form a polypeptide. And then there'll be hydrogen bonds between the polypeptide to either form an alpha helix or beta pleated sheet. And then after that forms, there'll be further coiling to form a more folded chain. So in the secondary structure, an alpha helix can be formed. So it's this ring-like structure, sorry, not ring-like structure. It's very spherical. I mean, that's not even a correct description. It's spiraling, basically. So alpha helix is due to hydrogen bonds between the oxygen atom in the CO and H in NH. So the amino acid, so for example, 7, this amino acid on position 7, there'll be a hydrogen bond with an amino acid which is 3 places ahead or behind it. So for example, 3 over here. So this will form a hydrogen bond. And 4 will form with 8 and 0, for example, which holds these structure tightly in shape. So basically hydrogen bonding can also form a looser straighter shape known as beta pleated sheet. So you give this type of structure during the in the exam paper, and then you'll be able to recognize them because they're straighter folded type shape. So this is of the same polypeptide. So for example, it goes like this, and then it'll turn here and then come back like this. So the hydrogen bonding is between amino acids which run parallel to it. So once that happens, so now we have the tertiary structure. So this happens when there's more bonds which are forming. So first of all, it's hydrogen bonding. So this is between strongly polar groups. So NH, CO, OH. So this is a hydrogen bond here, which is between the hydrogen to oxygen. And this is between polypeptide chains. So disulfide bonds form between these cysteine molecules. So cysteine is an amino acid which has this SH group as a side chain. So it loses hydrogen and then it'll form a really strong covalent bond between the two sulfur molecules, two sulfur atoms. So ionic bonds, so basically we know that amino acids have this NH3 or COH groups, right? So they can be ionized to form NH3 and CO minus, and then they'll form a ionic bond. Okay, so the next part that we need to know is that there's hydrophobic interaction. So basically, if there's two non-polar groups which come together, so there'll be hydrophobic interactions because the groups tend to stay together because they repel by the watery environment next to them. So if an entire protein is heated, the S covalent bond S bond will break the last because that is the strongest bond, and comparatively all the others are weaker, much weaker. So let's talk about the quaternary structure now. So the quaternary structure is basically a 3D arrangement of two or more polypeptides, or of a polypeptide and non-protein component, which is known as a prosthetic group. So this is in hemoglobin, for example. We talk about hemoglobin in a bit. And so proteins can be either globular or fibrous. So globular proteins, they have a role which is part of metabolic reactions. They curl up because they have water soluble properties because they have hydrophilic and hydrophobic groups. While fibrous proteins are not exactly soluble and they have more structural roles. And we talk about the example of collagen in a bit. Okay, so proteins. So the first thing we talk about is hemoglobin. So we know hemoglobin to be the oxygen-carrying pigment in red blood cells. It is a globular protein, and it has a quaternary structure because it has 4 polypeptide chains. And these 4 polypeptide chains are alpha globin and beta globin. So it can be either two, so it's not either, it's 2 alpha globin chains and 2 beta globin chains. And this hemoglobin molecule will fold because of hydrophobic R groups and hydrophilic R groups. So hemoglobin does have water-soluble properties, and the fold due to bonds form this specific 3D shape, and each polypeptide within the hemoglobin molecule will contain this thing called a heme group, and the heme group is a prosthetic group because it's not made up of amino acids. And this prosthetic group will contain an iron atom. So this iron atom is what allows hemoglobin to carry oxygen. So it binds to one oxygen molecule. And since there's 4 heme groups in one hemoglobin molecule, 8 oxygen atoms can be carried at a time. And hemoglobin binds to oxygen to form oxyhemoglobin. So this is what I'm talking about. This is the simplified structure of hemoglobin. So there's this 2 alpha chains, 2 beta chains, and a heme group is present at each polypeptide. So collagen is an example of a fibrous protein which plays a structural role, and it has a quaternary structure as well. So collagen molecule is made by 3 helical polypeptide chains. So these 3 helical polypeptide chains will wind around each other to form a triple helix. And this triple helix is held by hydrogen and covalent bonds. So in this helical polypeptide itself, each third amino acid in the polypeptide is glycine. So as we discussed previously, glycine has an hydrogen atom itself as R group. And this hydrogen atom is basically a very small R group, which allows compact packing of 3 polypeptides. And then collagen molecules can form cross links with other collagen molecules that run somewhat parallel to it. to form fibrils. And just take note that the end of the parallel molecules are staggered. So I'll come back to what it means that it's staggered in a bit. And these fibrils will lay alongside each other, and they'll also be held in bundles by cross links to form fibers. So this provides high tensile strength. So there's multiple bonds, there's strong covalent and hydrogen bonds. There's hydrogen bonds holding the fibrils together to form bundles. There's holding collagen molecules together to form bundles. So it gives this high tensile strength, and it allows it to be flexible as well. So what does it mean that the collagen molecules are staggered? So notice how these molecules, right, they are not in a straight line. So these gaps between the molecules, this gap, for example, here, it's covered by this thing. So it prevents weak spots from arising. So for example, if it all was neatly arranged, for example, all of these gaps were lined together, there would be a weak spot running across this collagen molecule, right? And in muscles, they can form a mesh. So it goes in different directions. So it helps to reinforce other collagen molecules. So last thing we need to talk about is water. And so we have somewhat studied the structure of water in chemistry, right? So it's a polar molecule. Oxygen is electronegative atom. So it can form hydrogen bonds between the lone pair of oxygen and hydrogen of another histo molecule. So between water molecules. So this hydrogen bonds are formed between water molecules. So first of all, it makes a good transport medium. So majority of our blood is made up of water. That's because a lot of substances can dissolve in it. So it's a good or excellent solvent for ions and polar molecules specifically. It also has a high specific heat capacity. So a lot of energy is required to increase its temperature. So this doesn't change the internal temperature. And why shouldn't the internal temperature be somewhat constant? So you have to try to maintain the internal temperature, right? Because first of all, there's a lot of enzyme reactions going on. And if the temperature in our blood fluctuated, the enzyme reactions would not be happening at the optimal rate. The proteins would be denatured. So it's not ideal conditions, right? So since high specific heat capacity, it helps to maintain an internal temperature. So the internal temperature doesn't fluctuate. So high latent heat of vaporization. So we have talked about this previously in homeostasis. I think majority of us have learned this in our previous syllabus. So a lot of energy is needed to be transferred to the water molecule because there's a lot of hydrogen bonds between the water molecule. So what happens is energy is transferred from the surrounding to water. So when there's energy loss from the surrounding, the surroundings will cool. So this is why when sweat evaporates from our skin, it helps to reduce the temperature of our skin or on the surface of our skin. And the last property which is important is that since hydrogen bonds are forming, there's forces of cohesion. So we'll talk about this more in depth in transport in plants, but I'll just like to mention it here as well. So it helps to travel in long unbroken columns through vascular bundles. So it allows for mass transport in the arteries, for example, in the xylem as well. And it acts as a very important reactant as well. So majority of hydrolysis reactions requires water, right? So digestion, breakdown of large food particles, and etc. So, okay, so I think that'll be the end of everything that we need to cover in this chapter. So let's quickly discuss some questions so we can test whether you have completely understood everything, right? So let's read this question. Solutions of 3 biological molecules are tested for sugars. The table shows the colors of solutions after testing. So you have solution 1, 2, 3. They're heated with Benedict solution. So it changes from blue to green to orange. So any color change indicates presence of reducing sugar. And blue means that there's no reducing sugar present. So over here, boiled with hydrochloric acid, neutralized, and then heated with Benedict solution. So what was this test for? This was a test for non-reducing sugar. And all of this indicate that non-reducing sugar is present if there was a non-reducing sugar present in the sample. So the question reads, which solution contained glucose before testing? Okay, so glucose is a reducing sugar. So solution 2 and 3, heated with Benedict solution show that there's a presence of reducing sugar. One is out. Blue means no reducing sugar. One is out completely. Option is D, and that's the correct answer. So this is my explanation if you didn't catch it. So glucose is a reducing sugar. So there must be a color change in Benedict's test. So one is wrong because the color remains blue, which means glucose is absent. So the answer is 2 and 3, which is D. So the next thing is a solution containing equal masses of amylose and amylopectin is completely hydrolyzed. So amylose and amylopectin, this is a mixture which is present in starch. And starch is a polymer of alpha glucose. Okay, so if it is hydrolyzed, what will be produced? Alpha glucose molecules, right? So this question asks which molecules will be found after the hydrolysis. So both of these compounds, amylose and amylopectin, is a polymer of alpha glucose. So beta glucose will not be present. So all of these B, C, D, all are wrong because it mentions that beta glucose will be present, which makes no sense. So the answer is A. So starch is a polymer of what? Alpha glucose. So amylose and amylopectin is made up of alpha glucose. So upon hydrolysis or breaking of glycosidic bonds, only alpha glucose will be present. So if you didn't understand my explanation while I was explaining it, just pause here during this slide which says answer and just read through, maybe you'll get it. So the next question, which feature of phospholipids enables for a bilayer to be formed in the cell surface membrane? So they're insoluble in water? Not exactly. They are polar molecules? No, because they have hydrophobic and hydrophilic parts as well. So C makes the most sense, and they may contain saturated, unsaturated fatty acid tails. So this is not really a feature that enables them to form a bilayer, but this will be a feature that you need to take into account for the fluidity of the membrane. So D is out, B is out, and A is out. So the final answer is C. So the next question, which molecules contain the following bonds? Ester, hydrogen, disulfide. So amylase is a protein. Hydrogen, hemoglobin, catalase. Catalase is also a protein. Okay. So read through the options, and I'll give you a second to just think about it yourself. Okay. So phospholipids contain the ester bond, which is between the head part, which has the OH, and the two fatty acid tails, which has the carboxyl group. Cellulose, hydrogen bonds are present between the cellulose molecules to form the microfibrils, and then the microfibrils to form the fibers. Disulfide. So where is disulfide present? Disulfide is present in the tertiary structure of proteins. So antibodies are proteins. So I had said that in one of the slides. So if you didn't catch that, do take a note here as well. Okay. So let me just repeat everything that I just said so you have a better understanding. Phospholipids have ester bonds, which is formed between head and two fatty acid tails. Cellulose, it forms glycosidic bonds, but more importantly, the question asks about hydrogen bonds. So between cellulose molecules and microfibrils. Antibodies, they are proteins which have a tertiary structure. And tertiary structure, they have bonds which are between 16 molecules, right? So they form disulfide bonds. So this concludes our lesson on biological molecules chapter, which is the second chapter of the AS biology syllabus. If you have any question, leave them in the comment section and I'll get back to you as soon as possible. I hope this video was helpful. I hope the other videos are also helpful and you're getting a better understanding of the subjects that we are uploading right now. And these videos are being very beneficial to you. And as usual, if you find the video helpful, do consider liking and subscribing the video, and do share them with your friends so more people can benefit. So until the next chapter, we'll meet, and happy learning to learn.